Well, welcome to Lecture 11 on molecular genetics. Now the next two lectures are going to be about genetics, and so the focus of this particular lecture is more on DNA and the components that make up the genome. And then we'll move on to looking at how psychologists, neuroscientists have used genetics to look at things like behavior and try to understand different traits, personality traits, and psychiatric disorders and so on. All right, so let's move on in this lecture. What I want to do is first of all introduce some terms, and then we're going to talk about the human genome. Next, we'll talk about DNA and its replication. We'll introduce the central dogma of gene expression. We'll look at the focus on transcription, and then we'll have epigenetics. Finally, in order to get ready for the next lecture, I'm going to give you some background to behavioral genetics by introducing just a little bit about Darwin's theory of natural selection and Mendelian genetics. Again, it's important to remember that these lectures that I'm giving you, like these ones on molecular and behavioral genetics, are meant to be accompanied by the Sapolsky book called Behave. And you'll see that in Chapter 8, this is where a lot of this material is covered. And also you'll want to look at Appendix 3 that does a quick overview of how protein synthesis and so on happens with genetics. All right, so obviously one of the reasons why we're talking about genetics here and looking at it in terms of social behavior is that everything is genetically influenced, right? So we're going to start with that premise that everything is genetically influenced. Maybe not by just a little bit, some things are influenced a lot. But then the question is, why is that? Why do we have so much influence from genetics? Well, genes influence biological systems. And if you kind of look at this one where we're looking at just trying to understand why does someone watch television, okay? You can see that we're saying that the genes don't directly encode for television watching. Instead, what they do is that various genes like gene 1, gene 2, gene 3, all the way up to gene GK would be different genes that all somehow together encoded to different biological systems. And those different biological systems in turn would lead to different behaviors or different characteristics of a person. And so you can see that in the blue there, we have things like their overall activity level, how much they seek thrills, how much they're interested in sport, and all of those things could be influenced by those biological systems. So in the end, genetics has a lot to do with influencing the biological systems that we in turn are interested in trying to understand specific traits or behaviors that would lead to a specific kind of social behavior. So that's sort of the big picture about why we're covering genetics in these next two lectures. Now, on to terms. Let's go ahead and talk about some important distinctions here. One is that you'll see a lot in these next two lectures discussions of phenotypes. And so a phenotype is a set of observable characteristics or traits of an organism. And keep in mind that these observable characteristics or traits can be fixed things about a person, but they might also be behaviors that they engage in. And so examples could be things like hair color, how one walks, things that they're allergic to, all right? So those would all be phenotypes, things that you can observe about this person. And so then we want to try to understand why do they have those particular characteristics or traits. And then the genotype would be the alleles, or another word for allele is a variant. So variants on the genes that the organism carries that happen in a particular gene or a set of genes. So the idea here is that a genotype is when you can actually see what the different alleles or variants are that the person's carrying for one kind of gene or a set of different genes. And the hope here is making a connection between the genotype and the phenotype, where the genotypes would contribute to the phenotypes, much in the way that I was just showing you about how TV watching could occur. But this could vary in how much. That is, you've got to remember that the environment's also contributing to the phenotypes as well. So genotypes don't purely only have to do with the phenotype. It could be the phenotypes being determined by things in the environment. All right, so that's phenotype and genotype. Let's move on to talking about chromosomes and genes and so on. You'll hear the term genome. So this genome is the complete complement of an organism's DNA. So each species could have a complete set of whatever makes up that organism, that species' DNA. That's the genome. Then we have cellular DNA is organized in chromosomes. So the individual bits of DNA are going to be organized in chromosomes. And then genes have specific places on those chromosomes. Most genes are not coded in continuous stretches. Turns out that a lot of genes are actually broken up into two different bits. And so you can see here in the example that we have a gene that's actually, one gene is actually made up of two different parts. It's separated by another part that isn't actually the gene. And so the two different parts would be the exons, and then the part that's in the middle is called an intron. And so that's just sort of an interesting variation on how genes are. They're not just going to be at one location. They could be in these multiple locations separated by introns. All right, so that's the genome and chromosomes, genes, exons, and introns. Let's talk about the human genome, which was fully sequenced in 2001. I remember I was at a meeting in Washington, D.C. in 2001 when the news broke that the human genome had been sequenced. I think people were expecting it any day now, and then all of a sudden when I was in Washington at this meeting, people were quite excited and passing around reports celebrating how the idea that the human genome had been fully sequenced. Now, we've known for much longer before 2001, that there were 23 chromosome pairs in the human genome. But we didn't actually know what the sequence of genes were on each of those different chromosome pairs. And it turns out that in the end, we now know that there's somewhere between 19,000 and 20,000 protein coding genes. So that means that these are genes that will cause transcription to happen, then translation to make different proteins that are needed by cells to do their actions. This is actually a very small fraction of the entire genome. So somewhere between 1.5 and 5% of the entire genome is actually made up of protein coding genes, which is really pretty small. It turns out that the rest of the DNA, the genes that are there, are actually non-coding RNA genes. You'll see things like transcription factors I'll talk about a little bit later in this lecture. Regulatory DNA sequences, introns, etc., are all making up these different parts of the chromosomes. So they're not just these protein coding genes. In fact, that's the smallest part of what the entire genome is made up of. Let's look at a specific gene, and this one's relevant to the last lecture, number 10, where we were talking about sex and gender. And you'll remember that chromosomal males who have XY chromosomes, they're the ones that are going to become males while they're in the fetus. And I mentioned a few times there in that lecture that you had to have the Y chromosome, and you also had to have this SRY gene. The SRY gene is the sex-determining region of the Y chromosome. That's how you get the SRY. And so it's on the Y chromosome. So this is only something that XY males are going to have. And it encodes testis determining factor, TDF. So what that means is that particular gene is going to allow the transcription of a TDF factor. And during development then, it means that you're going to get the testes and the testicular hormones being created as a result of that TDF factor being encoded. Okay, so the TDF is going to lead to the testes, and then the testes themselves start to create, for instance, the testosterone and the other androgens, and so then you get the masculinization of the fetus, and so this makes a fetus develop as male. And again, you can look back at that last lecture where we talked all about that. That tells us then that actually the default pathway, if you didn't have the SRY, you didn't have the Y chromosome, is that it would actually mean that the fetus is going to develop as a female. So it's basically, it's this encoding in the SRY of the TDF that causes us to have males and to develop into males in the fetus. Okay, so that's just one example of a gene that's there on the Y chromosome. Now, genes themselves are basically telling us about what's underneath them is their DNA structure, right? So we have this DNA structure, the double helix, this double-stranded molecule that's analogous to a spiral staircase. And we know that there are like these side rails that are made up of deoxyribose phosphate chains, and then there are these base pairs that link, or the steps that connect between them that are linked by hydrogen bonds. And then what's making up those steps are purine bases and pyrimidine bases, okay? And the purine bases have a cell structure or a molecular structure that has like a double ring to it, so that's why they're called double ring purine bases, and that would be adenine and guanine. And then we have the pyrimidine bases, which would be cytosine and thymine. So it turns out that adenine always binds with guanine, and cytosine is always going to bind with thymine. So you can see the way that the colors are in this little representation here. So green's always with red, the light green's always with purple, and so on. And so that kind of keeps happening up and down along the DNA strand. So the next point that I want to talk about is how we get DNA to replicate. So how you get the DNA to replicate is you're going to take this double-stranded DNA molecule, and then each DNA strand is going to hold the same genetic information. So in fact, because each G can only bind with C, each T can only bind with an A, if you only have one half of each pair, you actually now know the entire strand, what the template is to make another strand. So what you can do is you can split that curving staircase there, and just with one of the two strands, that becomes like the parent or template strand, and then a daughter or new strand is going to be assembled from nucleotides. And so what happens is another strand is going to be created that matches up, so that if that strand has an A, the only thing that go into there is a T, and so on. And then what you end up getting is a resulting double-stranded DNA that's identical to the original parent DNA strands. Now, I'm really going through a lot of things here in molecular biology that we could cover in a few weeks rather than just in 10 minutes. So keep in mind, I'm just going to be just trying to give you the highlights of how this all works. And you could always go, if you want, and learn more about these things or read up on them on Wikipedia or whatever. But then the question is, why is this even happening? Why do we have DNA replication? And why would we have DNA replication? It's because cells divide, right? So especially there in the fetus, as the brain is developing, we're getting a spinal cord, the entire body is developing, cells are splitting all the time, right? We're creating more and more cells. And you might remember all this stuff about mitosis and meiosis, right? But anyway, during that process of doubling the cells, you're going to go ahead and get replication occurring prior to cell division. So that means that prior to the cell splitting off into two different cells, that daughter cell that's going to be created from the parent cell is going to need a complete copy of the original cell's DNA. And so that's why this DNA replication takes place. Now, occasionally we have mistakes in the replication process. It's very, very rare, but this is what we talk about when we talk about mutations in genes. So there's some sort of change in the nucleotide base sequence of the genome. This is rare though, all right? But the problem is if that does happen, there's some sort of mix-up here in terms of the way this all happens. That would mean then that you'll get the wrong amino acid that will result in a non-functional protein due to improper folding. So that problem of a mutation means that things, when it gets transcribed later on, because you don't have the right sequence there's been something messed up. There was a mistake made in the duplication, in the replication. That means you're not going to be able to get the right protein that might be needed for a cell to grow or to have some other function that's done with that particular cell. This is almost always bad news, but sometimes this leads to a protein having a novel property that improves the ability of the organism and its descendants to survive and reproduce. So sometimes, these little mutations can actually lead to something that helps that organism in its new environmental niche to survive, and then we would pass on and have its descendants also have that mutation. So that's how mutations occur, largely through the replication of DNA. Let's move on to the fourth point of today's lecture, which is the central dogma of gene expression. That's just what it's called. And this has to do with the transcription and translation, how we make proteins from the DNA. Okay, so the overall goal here of transcription and translation is that the cell has some need to have something done. Something functionally needs to change. Maybe structurally something has to change in the cell. And as a result of that, it has to go to that sort of textbook of what the entire plan is here, the DNA, and find out exactly what it says about how to get, for instance, more glucose into the cell, or where it's supposed to be sitting as a cell related to other cells. And it does this by reading from that book, from the master book of the DNA, and then taking little pages out of it and making copies using proteins. And so, when it makes that copy, then it's going to go ahead and make a protein within the cell, in the ribosome. And the ribosomes make these proteins that then the protein themselves can maybe be a neurotransmitter, it could be a hormone, it could be something to do with the cellular wall. but the proteins are basically the basis of what all of these new products are. They're going to be created by the ribosomes. So transcription is the first step of gene expression, right? So this central dogma. And what this means is this is the process by which a DNA sequence is copied to produce a complementary mRNA strand. So this is going to sound like DNA replication, where we had nucleotides that would actually form along a part of the DNA. But instead, what's happening here is we're going to have something called RNA, an RNA strand. It's messenger RNA, so that's why it's called an M. So in other words, it's the transfer of the genetic information from the DNA into the mRNA, okay, into the RNA. So the DNA is the sort of like the main textbook, and so now this main reference is now going to be copied. Pages of it are going to be copied here with the mRNA strand. But like I said, it's like replication, but instead what we're doing is we're making RNA rather than a duplicate set of DNA. So this is the beginning of the process that ultimately leads to the translation of the genetic code via the mRNA into a protein. So that's the first step, is transcription. We're going to come back and talk a little bit more about transcription. Turns out it's probably much more important than we ever realized. Now, translation is the second step of gene expression, and the details here really start getting very particular, and I really don't want you to know all the names of this. I'd just rather have you understand what translation is conceptually, so you don't have to worry about all the things you see on this particular slide. It's getting really, really detailed now. But basically, keep in mind that ribosomes make the proteins from the messages that are encoded in the mRNA, right? So the ribosomes make these proteins. And so the mRNA has now made its transcription. It now knows, what the set of details are from the master plan. And then what's going to happen is these genetic instructions for a polypeptide chain are written in the DNA as a series of 3 nucleotide words. Okay, so in the original DNA, there were genetic instructions for a polypeptide chain that were basically 3 nucleotide words. And so these little bits are then going to be taken by the mRNA and brought to what's called the tRNA. And the tRNA are like the transport RNA. They basically take this messenger RNA and take those 3-letter nucleotide words and pass it on to the ribosome. So the ribosome knows exactly what the recipe is for making this particular protein. And so on the mRNA, this 3-letter code is called a codon. And then on the tRNA, this transport RNA is called an anticodon. And what's interesting is that in the RNA, it turns out that we don't actually have the T. In fact, every time there is a T in the DNA, it's replaced by something in RNA called a U, or uracil. Just a little slight deviation. It's the only thing that really changes, but every time you would expect a T, you get a U, okay? And then this is actually the genetic code, these little 3 nucleotide words that tell us or tell our ribosomes what to do in terms of making proteins. Okay, so that's translation, the second step of gene expression. So by the time it gets to the ribosome knows what to do. It manufactures the right protein that's going to then cause, like I said, a neurotransmitter to be formed, or maybe it's the way that the cell needs to be structured, whatever, okay? So going back through these last few slides, replication, transcription, and translation. Remember that to make new DNA, like for a new daughter cell, you need to do replication. So you make a copy of the genetic material. It's called replication. So when you think replication, think duplication, okay? Making RNA means that you're going to transfer the genetic code, the DNA, to the RNA. It's a transcription. So you're not taking the entire DNA sequence. It's not all necessary. It's only the parts that are necessary in order to make this, this code for the protein. So think of a medical transcriptionist copying the physician's words into another format. And that's what making RNA is in transcription. And then finally, the last step is we want to make proteins from those RNA transcriptions. So to make proteins, you have to have translation. So you think of how translation sort of relates to languages. The translation of biology translates the DNA information into new proteins that are needed by the cell. So again, a dense amount of information, I know. A lot of stuff that would be covered in multiple lectures if you took a course in biology on all of this. I'm just trying to give you the highlights so you can get the big picture. And of course, you can go back and look at Sapolsky for more edification on these different points. Now, sticking with sort of Sapolsky's approach to all of this, let's focus on transcription, because transcription turns out to be very interesting both from an evolutionary point of view and also from an individual point of view. Now, why is transcription so important? Well, transcription factors regulate genes. And what regulates the transcription factors? Well, it's the environment. So the environment regulates the transcription factors, and they in turn regulate the genes. So what does this mean? Well, the short stretch before each gene is called a promoter. Okay, so it's this little stretch of where the actual gene is that you would want to be able to use to create a protein, right? So just before it though, like a little part of that before it is called a promoter, right? Or you can see here what it says in the drawing is a binding site. This is sort of like the on switch for transcription. So when transcription of that gene is going to start, it has to be that you have this transcription factor that binds with that promoter area. So what turns it on? Well, a transcription factor will then bind to the promoter's binding site, and then transcription starts. And it turns out in order to bind to that promoter's binding site, like how does the cell know when it's supposed to start going ahead and doing transcription? Well, the environment, both internally and externally, regulates those transcription factors so they want to actually go bind with whatever part of the gene, part of the DNA is necessary. So both internal and external environments regulate these transcription factors. And a couple examples that are talked about in Sapolsky's book is, for example, a cell might need more glucose. So there's an internal environmental cue. And so a transcription factor for this would then bind to a specific promoter as a result of the cell needing more glucose, which would then activate a gene, which then codes for glucose transporters. And so you get the mRNA, and then the mRNA then translates at the ribosome into getting some protein. And so in the end, you get then some change, which is going to cause glucose transporters to actually be created along the membrane, and thus more glucose is transported into the cell. Another example that's more even further external from the cell itself would be, for instance, a mother smelling her newborn. And so receptors in the nose would activate a transcription factor in the hypothalamus. So the transcription factor is basically reacting to this smell. This is actually quite a few steps along this process. But those transcription factors in the hypothalamus would then lead to the production of more oxytocin, because the transcription would happen at that gene for the proteins that create more oxytocin. Okay, so transcription is really important in terms of these factors causing copies of the genes to be made, and then having the proteins being made that are accordingly. But the environment is really what triggers or sends off those transcription factors. Now, some more complications about this is that different transcription profiles result in different transcription factors being needed. So it often turns out it's not just one gene at a time that's going to be needed for any particular protein to be formed. That probably might surprise you, because you might think that it was just all the way I was presenting it so far, is that just one gene leads to one kind of protein. But it turns out, in fact, it could be 2 or 3 different genes work together. You have to have the information from 2 or 3 genes to get a particular protein that's necessary for the cell to do its thing. So, for instance, if we had 2 different genes, they would each have their own transcription factors, right? To say, turn on transcription of this gene, and this one also has a transcriptor factor. But if they need to be transcribed together for whatever purposes, because the environment needs both of those genes now to be activated, there would be a third transcription factor that basically would encode that these 2 both need to be transcribed. So the transcription factor of the third is a third type of template that basically says transcription's got to happen at both of these different genes. And of course, this gets more complicated with more and more genes needed. And it turns out that each transcription factor is coded by a gene itself. In order for us to have transcription factors to be created in the cell, you got to have a gene that tells us that that transcription factor needs to be created. And so much of the genome turns out to be actually genes for transcription factors. So we're back to that whole problem, like why is so much of the genome not really about coding for the proteins? Because so much of it is about this regulation of what's going on with transcription and other aspects of the genome. Sapolsky says, the more genomically complex the organism, the larger the percentage of the genome devoted to gene regulation by the environment. So if you again remember that the transcription factors are really being regulated by the environment, changes in the environment, both internal and external, cause changes in those transcription factors to become binding with that stretch just before the gene. That means then that because if you have a more complex sort of organism, there's going to be more of these transcription templates that involve lots of different genes at once. And so, in fact, a lot of gene regulation is going to become much more complicated. It's going to require a lot more of the genome just to be devoted to that gene regulation. Sapolsky also makes the point that evolution seems to be largely working on the gene regulation about these transcription factors rather than the genes themselves. So, having our ancestors pass on genes to us is, of course, very important. But another thing that they've done is they passed on the information about those transcription factors that were useful for different kinds of environments that helped them live long enough to be able to have descendants. And so you could say that evolution is actually much more focused on those transcription factors than the actual genes themselves. All right. Epigenetics is the next topic here. And so epigenetics is something that's been talked about in previously in the course, but basically these are changes in an inheritable phenotype that do not involve alterations to the DNA sequence. So again, what this means is you got a phenotype, some trait, some behavior that's heritable. Okay, something gets passed on from the parents to the child, all right? And then that phenotype changes, even though the DNA is there to tell it how it's supposed to behave or what the trait's supposed to be, but you don't get that. You get some change in the phenotype. And this doesn't involve alterations to the DNA sequence. How could that be? There's no mutations. There's nothing happening to the DNA, the code itself. But what actually happens is parts of the DNA could shut down, be turned off, so that when transcription is starting to happen, the transcription of that particular gene doesn't happen because of epigenetics. Now, there are many different epigenetic mechanisms. You can see in this drawing that I found on Wikipedia that some of those would include things that just happen normally in development, in utero and in childhood. It could be environmental toxins, chemicals, drugs, pharmaceuticals, just the natural aging process, what's in your diet. So all of those things can have a role in playing on basically shutting down parts of your DNA so that transcription can no longer happen. So the basic idea is that something happens to cause the transcription factors to no longer bind to the promoter. And there's lots of different ways this can happen. Some of them are illustrated here. For example, you could have like a myelin sheath that gets passed right around the place where the binding site would be for transcription. And so then the transcription factor comes along there, can't bind because it's been shut off, it's been wrapped up, all right? And so when this happens, transcription can no longer happen, and then that means that the gene is silenced, that the resulting gene that would depend on that transcription factor turning on gene activation doesn't do anything because there's no transcription factor to set the whole process off. So there's no transcription, no mRNA that's going to be made from that gene, no protein will be created as a result of that. Now, this kind of epigenetic phenomenon can happen for short periods of time. It might be something that lasts the organism's lifetime. And it's now been found that even this kind of thing happens across multiple generations, where some epigenetic mechanisms can be inherited, passed on down, so that particular gene no longer does its thing the way it might have done in previous generations. So it's a very fascinating area of molecular genetics to look at epigenetics, and it's just, again, something to keep in mind when we're trying to understand what's going on with our genetics in terms of our behavior. So this is a good place to bring up some take-home points from my lecture, and also just to borrow a little bit from Sapolsky's Behave here, to remember that genes are not autonomous agents commanding biological events. As Sapolsky says, they don't determine much. Basically, they're really very much affected by what's going on in the environment, and so they don't just override environmental concerns. They're in fact very sensitive to the environment. Genes are regulated by the environment, which includes everything that happens inside the cell, all the way to anything else that happens out into the universe. Much of your DNA turns environmental influences into gene transcription. So remember, this is the heart of where a lot of the environmental influences happen, is with gene transcription, rather than the coding for the genes themselves. Evolution is heavily changing the gene transcription rather than the genes themselves. And then epigenetics can allow environmental effects to be lifelong or even multi-generational. So there's some key things to take away from what we've talked about so far in molecular genetics. Now, as I finish out this lecture, the next thing I wanted to do is get us prepared for the next lecture, which will be on behavioral genetics. So to understand behavioral genetics, I want to just give you some background that led to behavioral genetics, then we'll stop and continue on at the next lecture with behavioral genetics. Now, we're going to, in lecture 14, talk a lot more about evolution, but I wanted to give some key points from Darwin's theory of natural selection as they pertain especially to understanding genetics and behavioral genetics. So keep in mind that one of the things that Darwin's theory of natural selection was looking at was how do new species emerge? That was like the central question that he was interested in, is why do we get all this variation over time and new species emerging? And there were three essential elements for the natural selection to operate according to Darwin's theory, because of course, how do new species emerge? Through natural selection. The three essential elements here are that there needs to be variation in the expression of a trait among members of a species. So this would mean that traits have to have some variation, like height can be varied. We could talk about behaviors that are variable. Some, there can be more aggressive members of the species and less aggressive members of the species. So there has to be variation in the expression of the trait. And that the variation itself must be heritable. That is, it has to be able to be genetically transmitted. And then third, that there's a struggle for existence among members of a species. And this is the idea that was promoted by Malthus, an economist or a political scientist, who basically says that the members of the species are fighting over a limited set of resources, right? And so there's going to be a struggle here in order to do this. I should just point out, by the way, for number 2, that genetics wasn't actually understood yet at the time of Darwin's theory of natural selection. He just knew that somehow this variation has to be heritable. And how it did that, there was no knowledge yet of molecular genetics, but it was understood that somehow the body did this, how we passed it on to our descendants. So the result of these different elements of natural selection is that individuals will have differential reproductive success, and you'll get gradual changes that will occur in a population in some species. And those gradual changes will shift the characteristics of populations, and new species will form from ancestral species. So that's the whole sort of like thing of the theory of natural selection in a nutshell, keeping in mind that Darwin could say all this without really fully understanding anything about molecular genetics yet. These are basic principles that would be later, the mechanisms for them could be then explained with molecular genetics. So if you put all this together and start to think about all the different pieces that are going into this, we have Malthus, who talks about competition for limited resources. We've got this important thing that variation in terms of breeds, races, and subspecies, according to Darwin, and that there are mutations, small changes in individual characteristics that could happen, right? And so that's going to lead to this whole idea of natural selection, survival of the fittest. And then when we get into understanding more about molecular genetics, we'll have an understanding of genetic variation, how the alleles of individual genes combine to give lots of different variation. And then the last thing that I want to talk a little bit more about in this particular lecture is just understanding Mendel's contribution to all of this. So he's the one that sort of first pointed out the mechanisms here without being able to see the actual molecular genetics behind it all. So let's just mention Mendel. Gregor Mendel was a Czech monk and a scientist who lived from 1822 to 1884. Now, he came up with these 3 laws of inheritance. He worked for many years with pea plants. That was what he chose in order to understand how these traits were inherited. He made numerous crosses with varieties of pure breeding pea plants, and then these varieties could be bred true for some characteristic each generation. And so he studied them very carefully, was able to have these pea plants grow pretty quickly, make little changes, breed them, and so on. And he presented his paper, we put all of this together, on experiments on plant hybridization at 2 different meetings of the Natural History Society of Brno in Moravia on the 8th of February and the 8th of March back in 1865. So these 3 laws that I'm about to present to you were talked about in these 2 meetings. But once the paper was out there, it had little impact, and it was only cited about 3 times over the next 35 years. It took a while for scientists to uncover what Mendel had already uncovered. So this is an example of the failure of obscure, highly original innovators to receive the attention that they deserve. Anyway, moving on, what are the 3 laws? So this is all stuff that, again, they learned from the pea pods, right? From these pea plants. So the first law is what he called the law of segregation. And so what he said was that each trait that you get interested in, it could be the color of the plant, the length of its leaf, or whatever the trait is that you're focusing in on, each trait is controlled by 2 factors which separate or segregate into the gametes. Now, what are gametes? Well, they're the genetic material, basically, that's being provided by each parent, each of the 2 original parents, right? And so you could have some phenotype, some characteristic that each parent is going to be contributing separately to the offspring. And so in this case, you can see that there was a white characteristic and a red characteristic that was in the parent generation. And then in the F1, the next generation of their offspring, you could see how many of them had that red characteristic or the white characteristic. And then you could continue to further follow this along by looking at the next generation after that. But the idea is that each time we've got the parents, each parent's going to have a gamete that's going to be contributing to the 2 different strands then of what makes up the genes in the offspring. All right, the second law. In Mendel's crossing experiments, he discovered that different characters or traits were inherited separately, that the traits themselves did not interact during inheritance. So this is what he called the law of independent assortment. So instead of them interacting, the traits were inherited separately or independently of one another. This is analogous to flipping 2 quarters, that the outcome of the toss of the first quarter resulting in heads or tails will not influence the outcome in a toss of the second quarter, or 20-cent piece, if you will, if we're talking about Australia. So the point here is that each of these gametes, what they contribute to its offspring, they have no dependency on each other. They're independently being provided by the father and the mother, and these 2 traits then are independent, being passed on to the next generation. Okay, so that means that it could be that which trait that gets passed on from the father or which trait that gets passed on from the mother just depends on basically like flipping a coin. And then we get to the third law, and this third law is called the law of dominance. And it turns out that one of the factors for a pair of inherited traits will be dominant and the other recessive unless both factors are recessive. So what we're saying here is that a parent the parents could be that the one parent could be tall and the other one parent is not so tall. And so the way you can see this is a capital T, a big T there for a tall, little t would be not so tall. And so the offspring is going to get a T, a big T, and it's going to get a little t. One offspring might get that by chance. And it turns out that the big T is the dominant factor there, right? The dominant trait. So if you have big T and little t, you end up being tall. And then you can see that another combination would be that the other big T from that parent could be combined with the small t of the other parent, and so on. So what's happening is that all of these parents, these combinations from a parent who's tall and a parent that's short, ends up giving us an offspring that's just tall, because tall is a dominant trait, dominant feature. Right? So all the offspring will have that dominance. Now, Mendel used uppercase letters to stand for alleles or variations that were expressed whenever they were in the genotype. So if he was talking about dominant alleles, he would use uppercase letters. And if he had lowercase letters, this would stand for the ones that are the recessive ones, that are expressed only in the absence of the dominant. These would be the recessive alleles. So true breeding tall pea plants carried these factors TT, whereas short pea plants always carried the factors t. And when Mendel crossed these varieties to produce the F1 generation, the resulting plants were all tall, and they all carried that genotype big T, little t. But then what happened in the next generation? So in the next generation, even though the phenotype of their parents was that they were tall, they actually had a genotype of big T, little t, right? So if a big T, little t were then to be paired with a short plant that's little t, that would mean that some of their offspring just by chance could be little t's, and they'll end up being short. So in that F2 generation, 3/4 of the plants could end up being tall, but 1/4 of them would end up being short. And it's just a matter of luck which gamete you get from your parent, right? Which of those characteristics, big T or little t, that you get when they get paired together, that causes this to happen. So the phenotypes are 3/4 tall, 1/4 short. And the genotypes would be 1/4 big TT, half of them would be big T, little t, and 1/4 of them would be little t. So that's 1/4 homozygous dominant, half of them are heterozygous, and 1/4 of them are homozygous recessive. Okay, so that's how the symbols are used and how you would look at that, and how psychologists ended up doing this when they started doing family trees and looking at characteristics across family histories. And so they started doing these things called pedigree diagrams. Now here's a joke pedigree diagram of how ice cream leads to all these different kinds of ice cream desserts. But in the actual real pedigree, it looks like this. So if you're going to make a pedigree diagram, you usually use a circle to represent a female. The square represents a male. You shade it in if you think that this characteristic that you're interested in, this phenotype, they have that trait. So this could be a psychiatric disorder. It could be something like blonde hair or blue eyes, right? And then a little circle with a line through it means that the person's deceased, or if they have a line through their square, that would mean they're deceased. Twins have their own notation in these pedigree diagrams. If someone's adopted, there's a notation for that. And if there was a miscarriage, which could be interesting for different reasons as well, you can indicate that there as well. So here's a pedigree diagram where you can see that we have a male who mated with a female, and then the female had this purple characteristic, this phenotype. And then we can look to see of their offspring, you can see that they had 3 girls and 2 boys. And it turned out that one of the boys had that same phenotype, and the other one didn't have the phenotype, but they had the genotype where they could possibly be a carrier for the trait, and so on. So you can see this combination. Actually, I should say that they only had, I should have said, sorry, that the parents had 3 offspring, 2 boys and a girl, right? And one of the boys would have gotten married or mated there with that female. That's what that is over off to the left. Over on the right there, you can see that one of the males mated with an unaffected individual. And so because this is a dominant trait, their offspring actually had that particular trait as well. All right, so anyway, that's just a little bit of how you would understand these pedigree diagrams. Note that these pedigree diagrams say nothing about genes, chromosomes, alleles, all that stuff we talked about at the beginning of the lecture. That's what makes behavior genetics different from molecular genetics, that behavior genetics could proceed for many decades without having to really understand things like DNA. Okay, so a lot could be said about inherited traits and trying to estimate how much was heritable and how much was due to the environment without understanding the actual molecular basis to genetics in the end. So we've now kind of completed all the parts to lead us to the modern synthesis in the early 20th century, where we're going to get the rise of behavioral genetics. And then we'll see that as science progressed in the 20th century, we then eventually get to have molecular genetics. And when molecular genetics comes into the scene, it sort of comes back and affects the way behavioral genetics is done. So in this lecture, we've talked about several different terms. We've introduced the idea of the human genome. We talked about DNA and its replication, the central dogma of gene expression, where we talked about transcription and translation. We focused a lot on transcription and transcription factors because of how the fact that the environment has such a big impact on transcription factors. We talked briefly about epigenetics. And finally, I've given you some background to behavioral genetics by going over some of the key points of Darwin's theory of natural selection and a little bit about how Mendelian genetics works. All right, that's all I have. And then we'll see you at the next lecture where we talk about behavioral genetics in full.